Our topic today will be a general background about the clinical laboratory instrumentation. This background will be separated into two parts. The first part will cover the first 2 points. The second part will cover the rest of the points. Prior to start with this background, I want to mention that a hospital is a dynamic environment which is changing continuously and rapidly due to the advancements in the technology. Also departments are considered as dynamic environments which are changing continuously and rapidly. To demonstrate this, I will talk about clinical laboratory. The workflow in a clinical laboratory can be separated into three phases: pre-analytic, analytic, and post-analytic. In the old environment, or in the old clinical laboratory environment, uh, only automation was related to the analytic phase, to the analyzers. We have different types of analyzers. We have chemistry analyzer, hematology analyzer, immunoassay analyzer, coagulation analyzer, blood gas analyzer, and so on. So the automation was only related to this phase, to the analytic phase, whereas in the new environment, the fully automated laboratory or lab, the automation is now related to the three phases: the pre-analytic phase, the analytic phase, and post-analytic phase. So now it's essential, in addition to the study of the analyzers, to study the automation in the pre-analytic phase and the post-analytic phase. Uh, in this functional block diagram shows different spaces and the circulation inside a clinical laboratory. The circulation and also the spaces have been changed due to the advancement of the technology, due to this new environment, which is a fully automated lab, which has made the pre-analytic phase and the post-analytic phase completely fully automated. Okay, so we need now in addition to study the analyzer, to study the automation in the pre-analytic phase and the post-analytic phase in order to understand this new environment and the needs for this new environment. In the next slide, we will have a small video which shows this new environment, this fully automated lab concept. As I have mentioned, all tasks in the pre-analytic phase, analytic and post-analytic are now completely fully automated. So as we see, the pre-analytic phase is now fully automated. Also, the analytic phase is automated, and also the post-analytic phase is also now fully automated. So we can see the different tasks in the pre-analytic phase, the different types of the analyzers: chemistry analyzer, hematology analyzer, coagulation analyzers, all them. And also we'll now see now the post-analytic part. So now we'll start with this background, as I have said before. We will start now with the first point in the background, which is the clinical laboratory. what is a clinical laboratory, what's the different sections of this clinical laboratory, and so on. So a clinical laboratory provides a diagnostic test data to aid in the detection, diagnosis, and the treatment of a disease. What are the responsibilities for a clinical laboratory? The responsibilities include correct identification, collection, and processing of patient samples, accurate performance of testing, timely reporting of the results, communication with physicians and other healthcare professionals. The workflow can, in a laboratory or in a clinical laboratory, can be separated into 6 main steps. The first step, the test is ordered. The second step, the sample is collected. The third step will be sample is delivered to the lab. All these steps are prior to start with the workflow inside the hospital, inside the clinical laboratory, which are the pre-analytic, analytic, and post-analytic. In the pre-analytic, the sample will be processed. In the analytic phase, the sample is analyzed, and the post-analytic phase, results will be reported. There are different types of samples or specimens. Most common laboratory samples type are the blood and urine. We have additional samples. We have the body fluids, sputum, stool, tissue samples, culture swabs, and so on. So why do we analyze our blood? For example, if I want to know the functionality or the physiology of kidney in case of a disease, or if it's working normally or abnormally, we make such test. If you want to study the condition of the cardiac muscles, or in case of the coronary disease, we make CK and LDH tests. In the case of delivery of liver diseases, we take the bilirubin. We have different types of bilirubin, which is direct and indirect, and so on. So the chemistry of the blood helps us to study the functionality or the pathology or the condition of a certain organ or tissue. This is an example of a medical examination overview where we start with an inquiry. We make an examination request. We send this for the laboratory. We make an analysis of the blood sample, and we give this result. As we said before, the work inside, or the workflow inside the laboratory, can be separated into 3 parts: pre-analytic, analytic, and post-analytic. In each part, we have different points which we should consider. As we said, the sampling, we have different types of samples which are analyzed by analyzers inside the laboratory. We have serum, plasma, blood, urine, cerebral spinal fluid, as well as different types of samples. The clinical laboratory can be separated, as we said before, into different areas. We have the core laboratory, which is constitutes about 73% of the volume of the whole volume or the complete volume of this lab. The core laboratory includes chemistry, hematology, toxicology, immunoassay chemistry, special chemistry, coagulation, and so on. The microbiology constitutes around 16% of the load volume. This includes blood, urine cultures, serology, and other areas. We have also another area in the clinical laboratory, which is transfusion services, which constitutes around 3% of the load volume. We have what the task include blood typing and cross-matching. In the chemistry section, we have also different, as we said before, we have different areas. We have the general chemistry. In general chemistry, we have tests like electrolytes, glucose, and other magnesium, calcium, cholesterol, and so on. We have also, in addition to the general chemistry test, we have the blood gas test. We have the partial pressure of oxygen in the blood, in the plasma, and we have the partial pressure of CO2 in the plasma, the pH of the blood, and other calculated parameters. All these are either measured or calculated in the blood gases. We have also the immunochemistry, which cover also different types of the test, including endocrine, specific proteins, tumor markers, and so on. And we have different sections as special chemistry, toxicologies, and other areas. In the hematology section, we have the CBC or differential. CBC is an acronym for the complete blood cell count. We have the coagulation test like PT and APTT and so on. We have the microscopy. In the microbiology, we have also different types of tests. We have the cultures, we have the blood culture, the urine culture, the sputum culture, wound culture, and so on. We have the antibiotic sensitivity, where we study what we call the MIC, the minimum inhibitory concentration. We have the serology, which is now these days very important due to the need to have a coronavirus test. We have a PCR lab where we make testing for the antigens and antibodies for the viruses. We have the viral antibodies and antigens testing. So we have different areas and sections, as we said before, in the clinical laboratory. We have chemistry, we have hematology, we have microbiology. The majority of the workload is in the chemistry area, which include different sections. Then comes the hematology area, and finally the microbiology area. Now we'll start with the second part, or the second point in this background, which is related to the composition and functions of the blood. The blood is the most important or mostly used sample to study the patient condition. So now we'll talk about the blood, about its composition and its function. Major components of a circulatory system, as we know, the heart, the blood vessels, and the blood. Usually in the study of the medical instrumentation in general, if we are talking about heart, we are talking about medical devices like an ECG machine, a defibrillator, a stress testing test, and so on, a patient monitor, vital signs, and so on. If we are studying the blood vessels, we are talking about medical devices such as the pulse oximeter, the pulmonary function testings, and so on. If we are talking about the blood, we are talking a great number or a large number of the medical devices, as we will see in the next, in the second part. We are talking about the analyzers, the hematology, the chemistry, the immunoassay, the coagulation. We are talking about the blood gas analyzer, electrolyte analyzer. We are talking about the blood culture, the PCR machine, extraction machine, and so on. So the medical laboratory instrumentation is dedicated to study the instruments or the medical devices which are related to the study of the different parts of the blood. The blood, as we will see later, is separated into cells and plasma, and we have different types of machine to study every bit or every part of this blood. So what is a blood? A blood can be separated into two parts, the plasma and the formed elements, or the cells. The cells can be separated into three categories: the leukocytes or the white blood cells, the platelets, and the erythrocytes or the red blood cells. The leukocytes also can be separated into two main categories: granulocytes and agranulocytes. The granulocytes are the neutrophils, basophils, and eosinophils. The agranulocytes are lymphocytes and monocytes. The plasma can be separated also into two parts: the serum and the coagulation proteins, and we will talk about this. So in the study of the medical instrumentation, we have different types of machines. We have, for example, the hematology analyzer, which is used to study the different types of the cells. We have the hematology analyzer to have analyze a blood sample. We have also another machines to study the different parts of the plasma, or for example, we have the electrolytes analyzer to study the electrolytes. We have also the black gas analyzer to study the gases. We have the chemistry analyzer to study the different chemistry constituents or different materials inside the plasma. We have also the coagulation analyzer to study the impacts of these coagulation proteins and so on. So each part of the blood, we have a different machine for it. So now, what is the blood? What is the plasma and serum? As we said before, the blood can be separated into two parts: the plasma and the formed elements. Blood is around 8% of the total body weight by comparison to the other fluids and tissues. This blood is separated into plasma and the formed elements or cells. As we said before, the plasma will be around 55% of the blood, and the formed elements or cells will be around 45%. The plasma, as we said before, also can be separated into two main parts: the serum and the proteins. The serum will be made mainly from water, which is around 91% of the plasma, 2% of solutes, and we have the proteins, which is around 7% of the plasma. As for the blood cells or the formed cells, we have the platelets, the leukocytes, and the erythrocytes. The erythrocytes are the red blood cells. The leukocytes are the white blood cells, which can be separated into 5 differentials or other differentials, as we will see later. So we have different medical laboratory devices to study every part of the blood. So we have a hematology analyzer to study the cells. We have a chemistry analyzer to study the components of the serum. We have a coagulation analyzer to study the proteins. We have an black gas analyzer to study the gases inside this plasma. We have an electrolyte to study the ionic constituents of the plasma or the serum. We have a coagulation analyzer to study the different types of the protein. We have different types, and we will talk about them in the later lectures or later chapters. So what is a plasma? The plasma is obtained when unclotted blood, as we see here, is centrifuged. The fluid above the cellular elements or the cells is the plasma. So in case we have unclotted blood is centrifuged, this blood will be separated into two regions. The above region will be the plasma. The down region will be, or the down part will be the blood cells. The blood cells can be separated into erythrocytes and a buffy coat, which is made up of leukocytes, which is W blood cells, white blood cells, and the platelets. So this is the plasma. So the plasma is the whole blood minus the cells, the blood cells, the WBC, the RBCs, and platelets. 90% of the plasma is made up of water. 7% are made of a plasma proteins. Most of these proteins are made by the liver or produced by the liver. 2% includes the solutes from different area. including, for example, the electrolytes, including organic nutrients, waste, respiratory gases, and vitamins. As we said before, to study this part or this part or this part, we have different type of the medical devices. In this diagram, we can see the ionic composition for different parts of the body. We have for the blood, we have for the interstitial fluids, the fluid between the cells. We have for the intracellular fluid. As we can see, the ionic composition for the blood and the interstitial fluid have a similar ionic composition. Between them is the capillary wall. So due to this different or slight different in concentration, we have the perfusion at the capillary wall, at the capillary level. This is the different of concentration inside and outside the cell. So this is inside the cell, this outside the cell, this inside the blood vessels, exactly inside the capillary. So we have talked about the plasma. What is a serum? Serum is a plasma from which is fibrinogen and other coagulation proteins have been removed as a result of clotting. So how we have a plasma? In order to obtain a plasma, it's obtained when a clotted blood is centrifuged. The fluid above the clotted blood will be the serum. As we can see in this photo, so we have a clotted blood, which is different from the plasma where we have unclotted blood directly or immediately centrifuged. In the case of the plasma, clotted blood is centrifuged. So serum is the liquid part of the blood here, which is after coagulation. The serum does not contain coagulation difference. So the main difference between the serum and the plasma is the coagulation factors. So as we said before, we have the blood, which is made up of the cells and the plasma. We have the plasma, which is made up of serum and coagulation factors. So this is the main difference. So why we need to study this? Because each medical laboratory device need different type of sample. For example, if we are talking about the type of sample for the chemistry analyzer, we are talking about the serum. If we are talking about the blood gas analyzer, we are talking about the whole blood. We are not talking only about plasma or semen. We are talking about the complete blood. If we are talking about the coagulation factors, we are interested in the plasma. So in order to make a specific test, we have a pre-analytic phase in order to obtain the specific type of samples we needed for this analyzer. So this is the main difference between plasma and serum. Again, serum is a plasma without the clotting factors. Serum is used for a chemistry analyzer, is used for an immunoassay analyzer, for electrolytes and other type of machines. Plasma is needed in the case of studying of coagulation factors using a coagulation analyzer, whereas we need the whole or the complete blood in case of the hematology analyzer. In order to have these different types of blood or different parts of the blood, we need a different pre-analytic phase. So this is important. For this, we're studying this. We're studying the serum, the plasma, and now we'll study also the blood cells. So in order to obtain the serum, as we said, we take or withdraw a blood sample inside a vacuum container. This vacuum container tube should have a clot activator inside it. We invert this tube 5 times. Then we leave it around 30 minutes to have a clot. Then we make the centrifuge of centrifuging for this vacuum container or a blood tube. and finally we get the serum. So now we take the serum and we we take a sample. For example, if we want to make analysis of a specific test in using a chemistry analyzer or immunoassay analyzer or even an electrolyte analyzer. So this is called the pre-analytic. So prior to start testing in a clinical chemistry analyzer or immunoassay analyzer or electroanalyzer, we need to have serum. Whereas in the case of the coagulation analyzer, we need to have instead of serum, we should have a plasma. In case if we want to have a sample to study using a hematology analyzer to study the different types of the blood cells, we need a complete sample without being coagulated. So we use different types and different pre-analytic procedure. This is very important in order to know, to understand the fully automated lab or the automation in the pre-analytic phase, additional to the automation in the analytic phase and the post-analytic phase. So now we'll talk about the blood cells. We started with the talk about the plasma, then we talked about the serum. Now we'll talk about the blood cell and their maturation procedure. All blood cells, if they are red blood cells or white blood cells or platelets, are returned to a stem cell, to a blood stem cell, which is found in the bone marrow. This blood cells is called hemocytoblast. This hemocytoblast will be first differentiated, then will be matured, finally will be functional and become a blood cell. For example, a red blood cell, a platelets, or a white blood cells. So each cell, each hemocytoblast blood stem cell will pass through three phases. The first and the second phase, which are the differentiation and maturation, will be done inside the bone marrow, whereas the functionality phase or life of this cell will be made inside the blood vessels or certain tissues. So all the blood cells are progenitors of hemocytoblast, the blood stem cell, which is a stem cell in the red bone marrow. This hemocytoblast will passes through three phases in order to become a blood cell, a functional blood cell, a red blood cell or white blood cell or platelets. So this life cycle will be separated into three phases: differentiation, maturation. These two phases are made inside the bone marrow, whereas the last phase, which is the functional phase, will be done inside the blood vessel or inside tissue. I will take an example: the red blood cells or the erythrocytes, to demonstrate this point. So inside the bone marrow, we have this stem cell, the hemocytoblast. First, a differentiation done. How is this differentiation done? We have a hormone which is secreted by the kidney. This hormone is known as erythropoietin. This erythropoietin will act into the hemocytoblast, makes it to differentiate in pro-erythroballast. This phase is called differentiation. This is done inside the bone marrow. The second phase will be the maturation, as we can see here. This also will be done inside the bone marrow. Then when it's become functional, it will become an RBC or erythrocyte, which will be released into the bloodstream. So as we can see, these are the different phases for each hemocytoplasm in order to become a blood cells. What we are currently analyzed using a hematology analyzers are the blood cells inside the blood stream. But sometimes, due to certain conditions, some of the cells before reaching the maturation phase, they are released into the bloodstream. So it's essential to know all this information to know what type of analyzer or hematology analyzer we are need. This also demonstrates the same point of view. We have the stem cell or hemocytoplasm inside the bone marrow, the stem cell inside this bone marrow, and we have the erythropoietin. As we can see, it's acted on this stem cells, and then it will be differentiated and later will be released after become mature into an red blood cell. Reticulocyte is a type of RBC which is not mature. It's an immature RBC. Sometimes it's released into the bloodstream in case of an urgent, in case of a blood loss or another conditions which needs an urgent release of these cells in the bloodstream to compensate the loss in the function of the red blood cells. So as we can see here, the red blood cells inside the capillary, they pass in a single file. As we can see here is also the function of the red blood cells. We are talking about the hemoglobins and their functional to transfer of oxygen to the tissue cells and the removal of CO2 from these cells, and the opposite inside the lungs. This is called, in both cases, we call this a perfusion. This is a perfusion in the tissue. This is a perfusion inside the lungs. So the difference is the CO2 will be removed from the tissue and oxygen will be supplied, whereas in the lungs, CO2 will be removed and oxygen will be supplied by the lungs. Here also an more demonstration about hemoglobin and about the hemogroups of this hemoglobin. Why we need to know this information? Because in the hematology analyzer we have different parameters which are need to be measured. One of them is the total hemoglobin. One of the parameters which are tested by the hematology analyzer is the hemoglobin or total hemoglobin. Here also the differentiation of the hemocytoplasm into neutrophil. So as we see, we have the granulocyte colony stimulating factor which acting on the stem cell to make differentiated, then it's become matured and then released. In this photo we see the different type of the granulocyte, the neutrophils, eosinophils, and basophils. The neutrophils are granules do not stain well with either acidic or basic dyes. Therefore, it's a neutral, and from this is coming the name neutrophil, which means that it's liking to be neutral. Where the eosinophil have an affinity, its granules inside the cell have an affinity for the acid dyes. So in order to stain the eosinophils, we need a stain with acidic nature and have affinity for the acid, the granules having affinity. Also the basophil, it's named because the granules have the affinity for a basic dyes. Also this information is essential to know because when we study the hematology analyzer, it's essential to know that to make this differential, to discriminate between the different differential of the white blood cells or the WBCs, we need to use different dyes, and we should know what type of dyes we need, if it's acidic or basic, or if being a neutral, to color a different type of cells and measured by the hematology analyzer. Here also another view for the different WBC as a 3D view. Also, this image is very important in the study of hematology analyzers, because we will have the differential part. The principle of the differential part is based on the staining of these cells, as well the use of light or laser to make a scattering and to discriminate between these different types. So when we know the real shape and the real parts of each cell, we can discriminate between them. And this is, as we said before, it's essential to make a design for a medical laboratory analyzer. Here also the platelets, which also is studied by the hematology analyzer. So the formed elements or the blood cells, we have different. We have the red blood cells, we have the platelets, we have the white blood cells. All are coming from the same cell, stem cell, which is the hemocytoplast, passed through three phases: differentiation phase, differential phase, maturation phase, and a final phase, which is the functional phase. The WBC or the white blood cells are cells with a nucleus. The RBC are blood cells without a nucleus, and the platelets are a portion of this cell. So this is the main difference, and this also is important to understand in case we to understand the principle of the hematology analyzer, where we make the discrimination between them based on the size of each type. The largest size will be for the white blood cells, which is cells with nucleus. The RBC will be smaller, and the smallest will be the platelets. All the size of these different part or formed elements of the blood will be in the range of micrometers. Finally, the functions of the blood. The blood have 6 functions: transportation, as we said, for transport of oxygen and the nutrients to the tissues, and the removal of CO2 and metabolic waste. Another function, which is the defense through the WC, the white blood cells. Temperature regulation is another function for the blood. The blood helps in the regulation of the temperature through deep veins and superficial veins, and this will be covered later in talking about a certain device, which is a vein viewer, which is also used currently in the laboratory. Prevent loss, hormone movement, and finally regulates the pH.